I'm so happy to be here to present our recent work. I'm Xiaoquan Su from China University of China. So today my talk is about our recent work named Phylo-Spec, which is a phylogeny-fusion deep learning framework that integrates multiple aspects of microbiome information for status recognition and disease classification. So we know the human microbiome plays a crucial role in maintaining human health. So we can make it as an indicator for the development of healthy status, as well as the development of diseases, especially at an early stage before the clinical diagnosis. So basically, due to the biological diversity and complexity, usually we sequence all microorganisms together by metagenomic shotgun sequencing or amplicon sequencing, and then we decode the short reads by informatical tools to know what microbes exist there and their proportion. So based on such profiles, we perform the diversity analysis to know the difference between groups of disease samples and health controls, and then use the data models like machine learning or deep learning to classify the status and diseases of new samples. However, the modeling of microbiome data is challenging. The one thing we should notice is the feature misalignment or sparsity due to the data heterogeneity, the algorithm inaccuracy for profiling, and sequence errors. So here I have an example. So we have two groups of S1, S2 as like health control and disease for training and modeling. And they have a very different distribution on the microbial communities. And we also have a new sample of S3, which is very similar to group S1. But unfortunately, there might be some that were mislabeled as other very close species, like here, feature shift. So in this case, S3 might be misclassified as group S2 because simply due to the inexact here. Recently, some approaches also integrate the tree-like hierarchy during the modeling, like the PMCNN or MetaDR or Deep Phylo. However, most of them failed to handle the unclassified species that is incompletely annotated by the lowest common ancestor profiling algorithm. So here is another example. We also have a S1, S2 as training, and a new sample S4, which is similar to the group S2. But it is possible that some microbes do not have a complete annotation, only on the genus or family level, like this unclassified A under the genus A. So here we cannot directly place unclassified A to any node in the tree because they belong to different branches in the tree. So most approaches just discard this part, and only with the remaining component, like here, the S4 can also be incorrectly recognized as group S1 here. So another problem is that the tree-like models can hardly measure the contribution of different microbes features for the classification of different diseases. We know that for some diseases like ASD or type 2 diabetes, only a small fraction of microbes associated with the change of status. So in this case, targeted detection with biological insight is limited. So here we propose the Phylo-Spec, which can capture the phylogeny relationships among the microbes with the taxonomy information to improve the detection performance. So in general, the Phylo-Spec has three key points. The first is the feature fusion in the phylogeny tree by a get convolution order algorithm. We post all the traversal in the tree, which can parse the information from the leaf node to the root node to release the effect of feature misalignment and sparsity. Then during the post-order traversal, we also dynamically map the unclassified microbes to some virtual node that are only corresponding to the sub-branches under the same taxonomy. So here, for the unclassified genus A, we set a virtual node of N7 prime that only contains the 3 branches under the same taxonomy about genus A. So fully use the incomplete information rather than discarding them. Of course, we also design an importance score by computing the information gun at the root, and then recursively propagate it through the whole tree to the children to measure the contribution for different microbes in the classification. So here to confirm and verify the effects of phylogeny aware and handling of unclassified species for this model, we use the 2 synthetic data sets. In data set 1, we simulated the distribution of the S1 and S2 groups, as I just mentioned, and artificially set some feature misalignment and feature drift in group S1. So from the beta diversity here on the left side, we can see that the regular distance metrics like Bray-Curtis or GSD mix the 2 groups by mistake. And on the other side, the phylogeny-based distance like Unifrac or Metastoms can distinguish the 2 groups as we expect. So this shows the importance of phylogeny information, diversity analysis, and the classification of different groups. So here the Phylo-Spec achieved an optimal performance of AUC like 0.94 that outperformed the other regular models. So in the dataset 2, we further made up to 20% of species in S2 as unclassified with only genus or family level annotation. So the performance of most other models decreased significantly, probably due to they dropped the incomplete information. And here the Phylo-Spec still can achieve the AUC near 0.9 because it used the incomplete taxonomy in the modeling. We also rank the contribution of microbes for grouping the 2 data sets and rebuild the model with only the top ranked species for noise cancelling. So here the results show that this effort not only improved the performance for the precise detection for specific disease, but also provide the explanation for the further diagnosis from the biological aspect. So we also apply the Phylo-Spec on real data set for the multiple disease classification across microbes collected from 18 different studies, even with the variation and inconsistent annotation among the cohorts or studies. The Phylo-Spec can still provide a balanced performance among different diseases with high AUC and kappa coefficient. So the source code of the Phylo-Spec is available via our GitHub repository. And here I want to thank my collaborators and all the contributing authors, as well as the founding agencies. that support this work. Thank you very much for your attention.
So we have time for a question. Okay, maybe I have one. So you showed all kinds of evaluations. So what are the most important metrics in your mind to assess the performance?
We actually evaluate the performance on different aspects like the AUC, RUC, AU, PRC, and the F1 score, and also we compute the kappa coefficient for the classification of multiple categories.
So you just think it's good to provide a comprehensive assessment across them?
Yes, we just want to make a fair comparison for the especially for the users who are not very familiar with the machine learning, because there's a lot of parameters we can adjust during the comparison, so we do not take too much on that. Just make some simple comparison.
Okay, thank you very much.
Thank you.
Let's thank him again.